We hope you enjoyed that short break. We will now continue on with our last two presenters presenting on topics recently reaching milestones. First, we will hear from Dr. David Strauss, who serves as FDA's Director of the Division of Applied Regulatory Science. Dr. Strauss oversees and coordinates FDA's research activities supporting the comprehensive in vitro proarrhythmia assay initiative, including the evaluation of ion channel pharmacology data, development and validation of an in silico computational model of the human ventricular myocyte, design and interpretation of induced pluripotent stem cell-derived cardiomyocytes assays, and assessment of novel ECG biomarkers in phase 1 clinical trials. And finally, we will hear from Dr. Alisa Vespa from Health Canada's Therapeutic Products Directorate. Dr. Vespa worked several years as a senior evaluator evaluating non-clinical data for new drug submissions. More recently, she joined the Office of Risk Management to work on the management of nitrosamine impurities in pharmaceuticals. She has also contributed to several ICH guidelines, including ICH S1, M7, Q3C, and Q3D. Please join me in welcoming our speakers.
Thank you for the opportunity to talk to you today about the ICH E14 and S7B Q&As that are on the clinical and non-clinical evaluation of QT or QTC interval prolongation and proarrhythmic potential. My name is David Strauss. I've been the rapporteur of the working group, and I'm from the FDA. The ICH E14 S7B Q&As were adopted by the ICH Assembly this past February. You can see a screenshot of the ICH Q&A document in the middle, and over on the right is a screenshot of the ICH website where, in addition to the Q&As, the concept paper and work plan, there is training material available on the website that includes a comprehensive PowerPoint deck and a supplemental file with tables of how you would go about including data to submit to a regulatory agency.
The problem that we're addressing is drug-induced torsade de pointes, or TDP, also known as twisting of the points. It's important to note this because QT prolongation gets a lot of attention, but QT prolongation by itself does not harm a patient. It's only when it proceeds to torsade de pointes, which can then rarely proceed to sudden death. In the 1990s to early 2000s, the community came together and asked, what do the torsade-causing drugs have in common? And it was recognized that they prolong the QT interval on the ECG, prolong the action potential at the cellular level, and block the hERG potassium channel at the cell membrane. In response, ICH developed the S7B guideline focusing on the non-clinical cardiac safety pharmacology to assess whether a drug blocks the hERG channel, prolongs the QT in non-clinical studies, and follow-up studies can be conducted as necessary. This has been successful in bringing investigational drugs forward into first-in-human studies. However, there have been issues that we'll come back to in a minute. In addition, with E14, the clinical guideline, it was established also in 2005 and describes the so-called human thorough QT or TQT study. It established a very sensitive threshold for ruling out TDP risk, approximately a 2% increase in QT, which is very small, and it resulted in it being the most intensive and expensive clinical pharmacology study. There have been multiple prior E14 Q&As that have been successful in preventing drugs with unknown TDP risk from reaching the market. However, there's room for improvement. The S7B studies inform safety before first-in-human dosing, but then are largely ignored, as shown by this large red line separating non-clinical and clinical. And the clinical assessment relies on human QT, which is an imperfect biomarker. It's sensitive but not specific. Prior E14 Q&As only allow for a thorough QT study substitution or waiver under narrow requirements, specifically very high exposure achieved in the concentration QT analysis. It often cannot be met. And the prior E14 Q&As also only allow for limited decision-making when a thorough QT study or equivalent cannot be performed. This results in unclear risk communication and labeling, such as having no large QT effects. In addition, E14 states that the TQT study is intended to inform whether ECG monitoring is required in all phase 3 trials, as not all QT prolonging drugs are proarrhythmic. However, in practice, drugs with a positive HERG or QT signal are often dropped from development. Thus, there's an opportunity. While at adoption, E14 suggested a QT interval evaluation independent of S7B results, both documents highlight the need for integration of information in a manner which is informative as a totality of evidence. Thus, our new E14 Q&As are directed at scenarios where the non-clinical data can reduce the number of clinical studies, streamline drug development, inform clinical regulatory decision-making at the time of a marketing application with non-clinical data, and inform labeling to better communicate risk. The E14 S7B working group, or initially discussion group, reached an agreement in 2018 on a two-stage approach. It starts with stage 1 Q&As. For S7B, these are on an integrated risk assessment, combining non-clinical and clinical, best practice considerations for in vitro and in vivo assays, and defining principles of proarrhythmia models. In stage 1, the E14 Q&As focus on the use of non-clinical data to inform regulatory decision-making in late stage of clinical development and at the time of a marketing application. The new Q&As to S7B, well, first, this shows the current S7B guideline as described in 2005. It consists of IKR and QT assay, core assays going into an integrated risk assessment. You can have follow-up studies if needed, and you get an evidence of risk. Our new Q&As focus on the integrated risk assessment considerations when non-clinical data are used prior to human testing and later in development for E14 scenarios as described in S7B. In addition, the new S7B Q&As also focus on best practice considerations for ion channel assays and in vivo QT assays, and best practice considerations for myocyte assays, including with induced pluripotent stem cells that can be used as follow-up studies, and the principles of proarrhythmia models that can be used to determine whether a QT prolonging drug is actually proarrhythmic or not. The stage 1 Q&As describe the two scenarios to use non-clinical data to inform clinical decision-making, and they focus on the so-called double negative scenario and the non-double negative scenario. The double negative is when you don't have HERG block and you don't have QT prolongation, and the non-double negative is when either or both are positive. In the revised E14 Q&A 5.1, we focus on the use of double negative non-clinical data to allow for additional TQT substitutes when the drug exposure in concentration QT analysis is not enough to meet the current requirement. For instance, if this is the requirement for a thorough QT study to reach the high clinical exposure scenario, to get a TQT substitute, you have to reach 2 times this high clinical exposure, and this often is not met. With the new Q&As, the TQT substitute with supporting non-clinical data will only need to reach the exposure level normally required for a thorough QT study. The impact is that this can reduce the number of clinical studies in drug development by substantially reducing the number of thorough QT studies, and it can affect a large number of drugs, as about a third of drugs QT studies fall under Q&A 5.1 in the past 5 years as submitted to FDA, and only about 40% of those cover the 2x high clinical exposure. With the revised E14 Q&A 6.1, we focus on the use of double negative non-clinical data to inform regulatory decisions and labeling when a TQT study or substitute cannot be conducted because of different examples like safety concerns with healthy volunteers, feasibility concerns in patients that result in a lack of positive control or inability to achieve high exposures, or confounded QT assessment. And the impact is that we can change regulatory decision-making and labeling, as these cases often currently result in a finding of no large QT effects, but with the new Q&A, a conclusion of low risk can be reached. This also has the potential to affect a large number of drugs, as about 25% of QT studies submitted to FDA fall under Q&A 6.1. What about Herg block and QT prolongation when they are present? I showed this slide earlier in the presentation, but it's more complicated than this. A drug that blocks Herg results in unopposed inward currents, sodium and calcium, and these can trigger the extra beats at the cellular level that can trigger torsade de pointe, the real concern. This top set of images shows what was on the last slide. When you block the Herg channel alone, you get QT prolongation that can lead to torsade de pointe. However, with balanced multi-ion channel effects, where you have Herg block and also have late sodium, calcium current block, or both, you can have QT prolongation that does not lead to TDP. And there can also be other non-ion channel mediated QT prolongation, such as from autonomic effects, that does not lead to TDP. We need to evaluate this with the non-clinical clinical integrated risk assessment leveraging proarrhythmia models. In stage 1, when Herg block and or QT prolongation is present, we describe in the Q&As on how the integrated risk assessment can leverage additional studies, and they can be performed to assess TDP risks and contribute to the design of clinical investigations and interpretation of their results, but they're subject to case-by-case evaluation. It's important to note the question of whether QT prolongation is still an issue in drug development. This shows data from FDA over the past 5 years of full clinical QT study reports submitted to FDA. This would not include drugs dropped from development that have a positive QT signal if it's seen in preclinical animal studies, if it's seen in phase 1 studies, or even a thorough QT study, and the drug is dropped from development before a full study report is submitted. 20% still prolong the QT interval, and they're divided among many therapeutic areas, although the two most common are oncology and drugs targeting the central nervous system. This highlights that QT prolongation still is an issue in drug development, and we need to determine which types of QT prolongation are actually proarrhythmic and which are not. Thus, in the original concept paper for our working group, we indicated that in stage 2 we proposed to provide details on how additional data can impact specific decision making, and that guidance on follow-up studies and assessments could be provided. It may include a combination of assessing multiple ion channels, proarrhythmia models, assays for mechanisms of QT prolongation beyond direct Herg block, and that ECG biomarkers can be used to assess the concordance of in vitro ion channel and clinical ECG effects, focus on markers of differentiating different types of ion channel block, specifically the J to T peak interval, and we would need exposure response analysis for QT and the other ECG intervals. And the guidance would include how they would impact late phase clinical trial designs, such as the intensity of ECG monitoring, and regulatory decision-making at the time of a marketing application, which impacts labeling, and we can have more clear communication about risk. In stage 2, we also propose to define how to define low-risk drugs that do not require detailed QT-focused clinical evaluation. Large proteins and monoclonal antibodies already do not require detailed clinical QT evaluation. And the question is whether we can expand this to additional areas like other therapeutic biotechnology products, drugs with low systemic bioavailability, potentially other areas, and each may require different considerations.
In summary, the working group recently completed Q&As for S7B on the integrated risk assessment, best practice considerations for in vitro and in vivo assays, and principles of proarrhythmia models, as well as E14 Q&As on how to use the non-clinical data to decrease the need for thorough QT studies and improve regulatory decision-making and labeling when a TQT study or equivalent cannot be performed. The working group is now evaluating whether to pursue the proposed Stage 2 Q&As on how to use proarrhythmia models and ECG biomarker data to inform decision-making and labeling for QT prolonging drugs, and how to define low-risk drugs that may not require detailed clinical QT assessment. In conclusion, while at adoption, E14 suggested a QT interval evaluation independent of S7B results, both documents highlighted the need for integration of information in a manner which is informative as a totality of evidence approach. These new E14 and S7B Q&As are directed at scenarios where non-clinical data can reduce the number of clinical studies and inform clinical regulatory decision-making at the time of a marketing application. These Q&As also outline best practices and principles for new and existing in vitro and in silico models that have the potential to enhance the prediction of risk for human proarrhythmia risk. Finally, consideration is given in the Q&As to the 3 R's, that is the reduce, refine, and replace principles of animal testing, in particular with the best practice myocyte assays and in vitro ion channel assays, along with the principles for proarrhythmia models that outline criteria, considerations for qualification of proarrhythmia models that can be computer or in vitro models that can be used as follow-up studies to evaluate the risk of drugs that may prolong the QT interval or block the hERG channel. And these alternative methods can be used in place of follow-up animal studies. I'd like to thank all of the ICH E14 and S7B working group members from my fellow regulatory agency colleagues and the industry consortia. Thank you very much.
Good afternoon. My name is Alisa Vespa, and today I'm going to provide an update to the ICH Q3D R2 guideline, which recently reached step 4 of the ICH process and is now being implemented across the ICH regions. The ICH Q3D core guideline provides a framework for the assessment and control of elemental impurities in drug products and recommends permitted daily exposure values, or PDEs, for 24 elemental impurities administered by the oral, parenteral, and inhalation routes of administration. The Q3D core guideline was adopted by ICH in December 2014. Subsequently, a revision was made to the cadmium PDE for the inhalation route of exposure, and this revision 1, or R1 version of the guideline was adopted in March 2019. During the time the parent guideline was being developed, it was suggested that it would be beneficial to create an appendix and develop PDEs for elemental impurities in drug products that are administered by the cutaneous and transcutaneous routes of administration, as this would close the gap and facilitate harmonization and implementation of elemental impurity limits for the most commonly used routes of administration. This led to the development of a concept paper describing this proposal, and it was endorsed by ICH in September 2016. The expert working group then developed the draft appendix, and once consensus was reached on the Step 1 draft document, it was submitted to the ICH Assembly and it was endorsed in 2020. Soon after, the draft appendix entered Step 3 of the ICH process, which consists of regulatory consultation across the ICH regions, expert working group discussion of comments received during the consultation, and document revision as necessary. Step 3 concluded at the end of 2021 when the expert working group reached consensus on the final appendix. The appendix was then submitted to the ICH Assembly and was adopted as part of Step 4 of the ICH process in April 2022. This version of the Q3D guideline, which includes the appendix, constitutes the second revision of the guideline and is coded as ICH Q3D R2. It is now posted on the ICH website and the link is available at the end of this slide. This slide outlines how the appendix is organized and includes the title of each section. I will now take you through the document and highlight the more important aspects of each section. I will also outline any notable changes that were made to the appendix as a result of stakeholder comments received during the public consultation. The background section of the document summarizes at a high level the physiological role of the skin and outlines how transcutaneous absorption of materials through the skin and into the systemic circulation is dependent on the properties of the skin, anatomical site, the nature of the chemical applied, and the characteristics of the application. Limited research has been conducted to evaluate the systemic absorption of elemental impurities following skin application. Of the available data, the skin represents a significant barrier to systemic absorption for various reasons, including, for example, the presence of metal-binding proteins, and systemic exposure is reported to be less than 1% for elemental impurities evaluated and in scope of the ICH Q3D guideline. Since for most elemental impurities, bioavailability data are lacking, a generic approach was used to establish limits for elemental impurities as opposed to an element-by-element approach. In regards to scope, the appendix applies to cutaneous and transcutaneous drug products intended for either local or systemic effects. To facilitate readability, the term cutaneous products is used throughout the appendix. The appendix does not apply to drug products intended for mucosal administration, topical ophthalmic, rectal, or subcutaneous and subdermal routes of administration. In response to the comments received during the public consultation, a footnote has been added in the scope to indicate that the Q3D guideline is not intended to provide recommendations for labeling of allergens. In this case, applicants should refer to regional guidance, recommendations, or best practices for managing and labeling allergens. Section 3 of the appendix is on principles of safety assessment for cutaneous products, which begins by summarizing at a high level a review of the literature in regards to both local and systemic toxicities associated with elemental impurities. To this end, there is no indication of local skin toxicity except for sensitization, which applies to nickel and cobalt. Similarly, there is no indication of systemic toxicity following exposure by the dermal route, except for thallium, which is readily absorbed through the skin. For the most part, there is very limited information on transcutaneous absorption of elemental impurities, and it was not possible to determine the percent absorption for each element, which would facilitate calculation of an element-specific PDE. For this reason, a generic approach was used, and cutaneous PDEs were derived by applying an upward adjustment factor to the parenteral PDE, which assumes 100% bioavailability. This adjustment factor is termed a cutaneous modifying factor, or CMF. I will take you through a sample calculation on the next slide so that you can see how the CMF is calculated and applied. For now, though, it's important to note that the application of the CMF does not apply to drug products intended to treat skin with substantial disruption of the basal cell layer of the epidermis, as observed in the case of skin ulcers or third-degree burns. This is because a compromised basal cell layer may facilitate direct entry of the elemental impurity into the systemic circulation. To derive a PDE in this situation, a case-by-case approach is recommended, and consideration should be given to using the parenteral PDE for the elemental impurity as a starting point. A second important item to mention is for nickel and cobalt, as these elemental impurities exhibit sensitization potential, an acceptable concentration limit was also derived to limit skin reactions in sensitized people. This will be further discussed in a few slides. Moving on now to section 4 of the appendix, which consists of the nuts and bolts of how the cutaneous PDEs were derived. This first bullet outlines the formula for calculating the cutaneous PDE. As mentioned, the PDEs are calculated by applying an upward adjustment factor known as the cutaneous modifying factor, or CMF, to the parenteral PDE. Therefore, the cutaneous PDE equals the parenteral PDE multiplied by the CMF. There are essentially 3 steps required to calculate a cutaneous PDE. The first is to determine the adjusted cutaneous bioavailability, which is abbreviated as CBA in the appendix. For all elemental impurities except for arsenic and thallium, a maximum CBA of 1% is assumed. However, to account for factors that can potentially enhance cutaneous bioavailability, an additional factor of 10 is applied, resulting in an adjusted CBA of 10%. The second step is to calculate the cutaneous modifying factor, or CMF. This is done by dividing the assumed parenteral bioavailability of 100% by the adjusted CBA of 10%, which results in a CMF of 10. So for all elemental impurities except arsenic and thallium, the CMF is equal to 10. The final step is to calculate the cutaneous PDE, and this is done by multiplying the parenteral PDE by the CMF of 10. Using cadmium as an example, the parenteral PDE of 2 micrograms per day is multiplied by 10, resulting in a cutaneous PDE of 20 micrograms per day. As mentioned, a CMF of 10 is applicable to all elemental impurities except for arsenic and thallium. In the case of arsenic, transcutaneous absorption is greater than most other elemental impurities at approximately 5%. For this reason, the adjusted CBA is 5% times 10 to account for various factors that can enhance cutaneous bioavailability, resulting in a value of 50%. The cutaneous modifying factor is calculated by dividing the assumed parenteral bioavailability of 100% by the adjusted CBA of 50%, which results in a CMF of 2. So for arsenic, the cutaneous PDE is calculated by multiplying the parenteral PDE of 15 micrograms per day by 2, resulting in a limit of 30 micrograms per day. Finally, thallium is highly absorbed through the skin and, for the purpose of the appendix, is considered to be equivalent to parenteral levels. Therefore, there is no need to calculate an adjusted cutaneous bioavailability, and the CMF is equal to 1. Therefore, the cutaneous PDE for thallium is equal to the parenteral PDE for thallium, which is 8 micrograms per day. Cutaneous exposure to nickel and cobalt is associated with the potential to induce skin reactions in already sensitized individuals. For this reason, in addition to a cutaneous PDE, a cutaneous and transcutaneous concentration limit, or CTCL, was also derived for nickel and cobalt. In regards to nickel, the dermal concentration limit of 0.5 micrograms per centimeter square per week was used as a point of departure for calculating the CTCL. This dermal concentration limit is applied to consumer products under the EU Nickel Directive in EU countries, and since implementation, the prevalence of nickel allergy has decreased. So how was the CTCL for nickel derived? To begin, the dermal concentration limit of 0.5 micrograms per centimeter square per week was converted to a daily concentration limit of 0.07 micrograms per centimeter square per day. Then it was assumed that drug product is applied to a skin surface area of 250 centimeters squared. Multiplying the daily concentration limit by 250 results in a value of 17.5 micrograms per day, which is then divided by an assumed 0.5 gram daily dose of drug product, resulting in a CTCL of 35 micrograms per gram. And this is the concentration limit based on once daily application. As a result of the comments received during the public consultation, the expert working group added some text regarding why 0.5 grams for a daily dose and 250 centimeters square for skin surface application area are considered reasonable assumptions for calculating the CTCL. In regards to cobalt, a similar concentration limit was recently derived to minimize elicitation of allergies. Therefore, the CTCL of 35 micrograms per gram is also applicable to this elemental impurity. Section 6 of the Q3D appendix is on product risk assessment, and this slide summarizes some points to consider. First, it is recommended that risk assessments be prepared following the guidance provided in the parent Q3D guideline. Importantly, both the cutaneous PDE and the CTCL must be met for nickel and cobalt since they exhibit the potential for sensitization. Since the maximum daily dose for cutaneous products is not always clearly stated, it is recommended to estimate worst-case exposure to the elemental impurity. In some cases, it may be important to evaluate the retention time of the drug product considering typical conditions of use. For example, shampoos are applied for a short duration of time, which may affect exposure estimates and could form the basis of a rationale to adjust the PDE. Finally, a control threshold can be applied to the PDE and the CTCL. This threshold is defined as a level that is 30% of the established PDE or CTCL, and it can be used to determine if additional controls may be required. On the other hand, if the total level of an elemental impurity from all sources in a drug product is consistently below the control threshold, then additional controls are not required. Section 7 of the appendix, which is the last section, summarizes in tabular format PDEs for 24 elemental impurities, the CTCL for nickel and cobalt, and concentration limits for the 24 elemental impurities which were calculated assuming a 10-gram daily dose of drug. As for next steps, the ICH Q3D R2 guideline, which includes the new appendix, has been finalized and is now published on the ICH website. The guideline will now be implemented across the ICH regions. This is my final slide, and I would like to acknowledge the ICH Q3D Expert Working Group, as the information presented here is a work product of this group. And with that, I would like to thank you for your attention.
Thank you. We will now continue on to our Q&A panel. If you have not yet had a chance to enter your questions into the Q&A chat pod, please do so now, and we will answer as many questions as time allowed. Joining us for the Q&A panel in place of Dr. Friedman is Dr. Alexey Khrenov, CMC Reviewer in the Division of Plasma Protein Therapeutics in the Office of Tissues and Advanced Therapies within CBER. His review responsibilities include reviewing CMC sections of marketing and investigational applications and supplements for plasma-derived and recombinant hemostatic products. So we will start off with questions. We will go by speaker. So the first set of questions will be for Jill Adelberg and Dr. Teresa Mullen. So the first question is, what effect has the pandemic had on ICH's work? If one of you would like to respond, that would be great. Thank you.
So sure, I'm happy to get us started, and that's a great question. As Teresa noted at the outset, ICH's harmonization efforts have been critical in speeding the development of vaccines and therapeutics to prevent and treat COVID, and in supporting regulators' ability to collaborate. While development of some new guidelines has been delayed as experts focused on the pandemic, we're really proud of all of our experts who have made remarkable progress in advancing ICH work. We're also really excited that a number of working groups will be meeting face-to-face for the first time in 2 years during the upcoming ICH biannual meeting in Athens. They'll be holding hybrid meetings, leveraging the value of face-to-face discussions with virtual participation during core hours.
I think the only thing, thanks Jill, the only thing I would add to that is that, of course, our groups, being very innovative people, are also kind of figuring out what are the best practices and ways to work most efficiently in an all remote mode, virtual mode, the last 2 years, and we'll be learning, I think, more about what is most effective in hybrid mode as well in the coming weeks. Thank you.
Thank you. Our second question for you both is, how will ICH approach regenerative medicine and innovation in cell gene technologies? For example, point of care, decentralized manufacturing platforms, genetically modified cell therapies, etc., given the varying regulatory schemes of members?
Another good question. As the questioner noted, different regions do have varying regulatory frameworks, and this generally means that a topic is not yet ready for harmonization. The most helpful discussions at this stage might be continued discussion and exchanges among regulators to identify topics for harmonization or convergence that are more feasible. These discussions sometimes occur within IPRP, which is the International Pharmaceutical Regulators Program, which is an organization comprised of ICH's regulators. And S12 is an example. The S12 Expert Working Group is currently developing a guideline on non-clinical biodistribution considerations for gene therapy products. Discussions on developing this guideline were advanced in IPRP first.
I just agree with everything Jill just said, and so I have nothing to add to that.
Thank you. We have one more question for both of you before we transition to some questions for Dr. Lin Yao. So the question is, can we know a tentative timeline for completion of all 5 steps of ICH guidelines?
So the time frame really depends on the subject matter and the availability of experts. As a general rule of thumb, ICH guidelines take about 3 years to complete, about 1.5 years for development of the draft, and about 1.5 years to develop the final harmonized guideline. But there tends to be significant variation. And for updates on development of any of the ICH guidelines, we'd really encourage folks to check the ICH website periodically. It's regularly updated.
The only thing I would add, I agree with everything Jill has said, and the one other thing to keep in mind is that I think what Jill's described is probably very accurately reflects what the groups have more control over, which is their work. Interspersed in there, of course, is the time period for collecting public comment, which is quite critical. So those time periods for public comment in different regions of the world can be a little bit varied. They're not necessarily synchronized, and so that can also introduce a longer total elapsed time frame across the all 5 steps. Over.
Thanks to both of you. We will circle back with you if we have time after we get through all the speakers' questions. So next we will be moving to questions for Dr. Lin Yao. So Dr. Yao, our first question for you is, who will initiate the pediatric extrapolation work? Per FDA advice, or the firm NDA itself initiate the pediatric extrapolation work?
Thank you for that question, and it's a really good one. You know, one of the things that we've learned is that it takes a long time to get pediatric therapeutics development completed. And on average, even in the United States, it takes somewhere between 7 and 9 years after adult approval before we see any pediatric labeling related to the studies that we've either required or requested. What we're trying to do is cut down that time. And the point of the pediatric extrapolation guideline is to figure out not only how to do extrapolation, but to incorporate the important need of collecting data that will support the extrapolation approach early in adult drug development. Because a lot of the similarities in terms of the disease and the response to treatment can be collected as part of the adult phase 1, phase 2 program. So I would say that, first of all, the pediatric extrapolation work doesn't begin after the adult approval. It doesn't even begin during adult phase 3 necessarily. We want you to start thinking about what the pediatric development program will look like, including the use of pediatric extrapolation, as soon as you've begun to think about how this drug would be used in adults and whether or not there's a use for it in children. And then the second part of the question, which is who initiates the work, I would say that is really initiated through conversations between sponsors and regulators. In other words, if you're developing a drug and you think there is going to be some potential use in children, that's at the point you should be starting to think about pediatric extrapolation. And that's at the point you should be thinking about talking to regulators about what that extrapolation approach would look like.
Thank you for answering that. We have another question for you, Dr. Yao. What are the minimum criteria to extrapolate the data to the pediatric population?
Right. So, I didn't have time to really go into the explanation of that very good question during my talk, but it is a good question. And the answer is that it depends. And I know that's not a great answer. But the idea is that we don't want to set minimum criteria. What we want you to do is look at all the available data, what data sources you have, and the guidance really talks about the types, sources of data, the quantity, quality of data that you could put together to support an extrapolation approach, and whether or not there are data that would support a particular type of extrapolation approach. So I don't want to say that there's a minimum amount of data, and that you should begin to think about whether extrapolation to whatever degree is acceptable at the beginning. There may be, in fact, even with a new molecular entity in a disease condition that we consider to be very similar, there may be an ability to extrapolate even with a new molecular entity. On the other hand, there may be diseases that we know very well that we might not consider extrapolation so acceptable if we know that there are differences that really have borne out in some of the programs that have already been completed. So hopefully that gives you an idea of the fact that we wouldn't look for a particular minimum standard.
Thank you so much, Dr. Yao. We have one more question for you before we move on to Dr. Khrenov. So your third question is, will there be additional training materials developed to help explain the concepts presented in the E11A guideline?
Right, and I mentioned we're going to try and have a case example training material because we recognize that the document is quite long. It's almost 50 pages long, and it's very dense. And, you know, it took us a while to figure out how to present it, and I think the table of contents suggests to you that it's a very comprehensive and detailed document. But we also recognize that there needs to be a way to operationalize that, and that just at the end of reading 50 pages, are you going to know what to do? So we've created, and we're about to, we hope, publish in the next month or so, a case example that you can refer to say, okay, this is what they mean, or this is how I need to operationalize constructing the extrapolation concept or putting together the study designs that would form the extrapolation plan. So we hope that through this case example, you'll get an idea, but we certainly have full intention of providing more detailed and several additional sets of training materials as we move forward to help explain different areas of the guidance. And certainly during the public comment period, if you think there's a particular section or a particular concept that would be benefit from additional training materials, please let us know through the public comment period.
Thank you for those detailed explanations. We will now be moving on to questions for Dr. Rick Friedman, and Dr. Alexey Khrenov will be responding to the questions for him. So our first question, I'm curious about acknowledging subjectivity. Would it be correct to assume that decisions which are not supported by metrics and data would be at a higher risk for subjectivity? And if so, does nothing in the background of the individuals involved in the QRM activity need to be noted? Does noting the background of the individuals involved in the QRM activity need to be noted in addition to the disclosure that the decision is not supported by data or metrics, as that might give some context as to the potential for this subjectivity?
Hello, everybody. I just want to explain first why I'm covering for Rick. I'm also the member of expert working group tasked with the revision of ICH Q9 guidance. So since Dr. Friedman couldn't be here today, he asked me to participate in these Q&A panels. And it's a great question, and it's sort of expansive, but I think the general idea captured really well. Subjectivity can indeed greatly affect the QRM process in the situations where data are limited, and actually bringing more data and proper knowledge management is one of the ways to control and reduce subjectivity. One of the biggest sources of subjectivity is, of course, personal biases and background, and I think that question is also correct in this regard. I wouldn't say that capturing subjectivity requires some sort of formal disclosure or note, because we're talking about people interactions, and certain level of discretion should be exercised in this case. However, definitely the backgrounds or biases should be acknowledged. What really helps in this situation, and it's actually mentioned in the guidance, is to have somebody who is experienced in facilitating QRM activity, somebody who is experienced in the process itself, in meeting dynamics, who can actually observe and acknowledge subjectivity and can try to control it. But in general, yes, I totally agree that noting the background of people, noting their biases, can really help to control subjectivity in cases where data are limited, which are, of course, mostly susceptible to subjectivity. Thank you.
Thank you. We have another question that's come in for you. When will the FDA publish the draft Q9R1 for comments?
Unfortunately, I cannot tell the exact date about this regarding the publication of revised Q9. I know that we're a little bit late compared to other jurisdictions, but we have apparently more complicated process. We worked really hard to move this guidance, and at this point, I expect that it would be done really, really soon.
Thank you. And we will take one more question for you before we move on to questions for Dr. Shabuddin. Another question for you is, you referred to training materials that will be released with the updated ICH guidelines. So how can we access those materials?
Well, obviously, these training materials to support the revision of ICH Q9 guidance, they're not ready. They're currently being developed. We are actively in the process. The expert working group currently working hard on the development of these training materials. We cannot tell when they will be available. However, as with other training materials for other ICH guidances, they would be available through ICH website. Whatever material the group designed, they will be posted at the ICH website. But again, I cannot tell when it will happen. Thank you.
Thank you, Dr. Khrenov. We will now move on to some questions for Dr. Shabuddin. The first question, do the Q2 and Q14 guidelines apply to analytical methods for biologics which are more variable?
Yeah, that's a good question. The goal of Q14 is to provide guidance for the development of an analytical procedure fit for the intended use over the life cycle for measuring an attribute, and with needed specificity, accuracy, precision over the reportable range. So, although biological methods are more variable, they should provide reliable, reportable results with specificity and reasonable accuracy and precision. So I think the concept and guidance provided in Q14 can also be applied to biologics for analytical procedure development. That's all I can say about it.
Thank you so much. We do have one more question for you. What is the relationship between ICH Q14 and ICH Q12 guideline on technical and regulatory considerations for pharmaceutical product lifestyle management and its annexes?
That's a great question too. The ICH Q12 guidelines, it's the guidance as the title says, technical and regulatory considerations for pharmaceutical product life cycle management. It provides guidance for a risk-based to facilitate the approach to facilitate the management of CMC post-approval changes in a more predictable and efficient manner across the product life cycle, mainly in the area of frequent manufacturing changes. Q2 describes tools such as risk-based categorization of post-approval CMC changes, for example, established conditions and their reporting categories, post-approval change management protocols, etc. The similar mechanisms are used in Q14 in the case of post-approval changes in analytical methods. Similar to ICH Q12, Q14 guideline describes principles to support change management of analytical procedures based on risk management, comprehensive understanding of the analytical procedure, and adherence to predefined criteria for performance characteristics. And knowledge gained from application of the enhanced approach to analytical procedure development can provide better assurance of the performance of the procedure. Also can serve as a basis for analytical procedure control strategy. There should be an increased understanding of the relationship between analytical procedure parameters and performance to facilitate identification of which factors require control, and thus enable a more appropriate set of established conditions and reporting categories. And this can focus on performance characteristics like specificity, accuracy, and precision. If a parameter is controlled through performance characteristics and criteria, that parameter may not necessarily need to be defined as an established condition or may be assigned a lower reporting category. So this approach provides an opportunity for more efficient regulatory approaches to related post-approval changes in case of analytical procedures in Q14. So these are the main similarities between Q12 and Q14. All the concepts and tools used in Q12 are used in Q14 as well.
Thank you so much. Thank you. We will move on to Dr. David Strauss. So the first question we have for you is, can you comment on whether sponsors are required to use the integrated non-clinical, clinical, integrated risk assessment described in the new E14 S7B Q&As?
Yes. Thank you very much for the question. So the new Q&As present options for sponsors to leverage high-quality non-clinical data in certain situations to either decrease the need for additional dedicated QT studies or to use as part of a decision-making package at the time of to weigh into decision-making at the time of a regulatory application on the risk of a drug. But these are, it's optional to use these newly elaborated described scenarios in the Q&As, and sponsors can continue to use the methodology and approaches previously described in, for example, the ICH E14 or E14 Q&As, the previously existing ones.
Thank you for responding to that. Your next question is, how can we learn more about how to implement the concepts in these new Q&As?
Yes. So with, as I briefly mentioned at the beginning of the presentation, we have put fairly comprehensive training material up on the ICH website. It includes a PowerPoint deck now in PDF form of about 90-some slides, but there's a table of contents, and you can go to the sections of interest. It contains a fair amount of detail, and in addition, there is a supplementary file that contains a series of tables that have hypothetical examples of the types of data that would be submitted when
you're going through one of these new integrated risk assessment pathways. So we hope that will be useful, and we anticipate more live training sessions that would be performed or carried out by some likely ICH training associates, that there'll be some sessions by some of the relevant professional societies coming up this summer.
Thank you. And we have one more question for you before we move on to Dr. Alisa Vespa. What triggers the QT-QTC prolongation, and how can one control it? Are all the prolonged QT-QTC drugs dangerous to the patient? What triggers the prolongation of QT-QTC in oncology drugs?
That's a very good question. So the most common reason for drugs to prolong the QT interval, in particular drugs that have a risk of torsade de pointe, which is the abnormal heart rhythm of concern, they block the so-called HERG potassium channel. And I discussed that in the presentation. And that is why when ICH S7B was put in place in 2005, it was required or strongly recommended that all drug developers should test their drugs against this HERG potassium channel. There are other ways to prolong the QT interval, having autonomic effects or some sort of confounded effects based on affecting heart rate. And not all of those methods of prolonging the QT interval are proarrhythmic that will cause torsade de pointe. And some of that is also due to drugs affecting other ion channels other than the potassium channel, affecting calcium and sodium channels. And so, yeah, not all QT prolongation is harmful. And in these Q&As, we have put in place principles for validating or qualifying proarrhythmia models that could be used to determine if a QT prolonging drug is actually harmful. And in oncology, it's really the same mechanisms in general, most commonly blocking this HERG potassium channel, but there can be other mechanisms because there's a higher sort of risk tolerance of oncology drugs, that these are life-saving drugs. There's more drugs that prolong the QT interval, but again, it doesn't necessarily mean that they are harmful and will cause torsade de pointe, and that's where putting into place these new principles for proarrhythmia models, we're hoping we can advance to have additional proarrhythmia models be qualified and fit for regulatory use.
Thank you, Dr. Strauss. We will now be moving on to some questions for Dr. Alisa Vespa. So Dr. Vespa, the first question is, what is the timeline for implementation of the ICH Q3D appendix?
Thank you for the question. As mentioned in the presentation, the Q3D guideline has reached step 4 of the ICH process and is therefore considered final. In regards to implementation, I can't speak to the FDA's implementation plan. However, on the Health Canada side, we are in the process of discussing the implementation plan for Q3D R2. It is likely that a timeline similar to the one used for implementation of the parent Q3D guideline will be utilized. Once the implementation plan is finalized, it will be communicated to stakeholders and also posted on the Health Canada website.
Thank you, Dr. Vespa. We have one more question for you, and that is, if an elemental impurity is present in a topically applied drug product and the level of the elemental impurity exceeds the permitted daily exposure limit, can the limit be adjusted if the drug product is removed from the site of application after a short duration of time?
Thanks for that question. So making an adjustment to a published ICH limit is considered case by case by individual regulators, considering both the rationale and supporting data provided. In the case described, a limit greater than the permitted daily exposure limit could be considered, and the rationale could be based on modifying the retention factor, since the drug product is only on the site of application for a short duration of time, whereas a retention factor of 1 is used for drug products that stay on the skin. The retention factor can be modified for drug products that are rinsed off the skin, as an example. Thanks.
Thank you, Dr. Vespa. And it looks like we have a little bit more time for questions, so we will circle back up to the top to the presenter list and ask some additional questions. So the next question is for Jill Adelberg, and the question is, do the same ICH guidelines and regulations apply to the medical device industry?
So the short answer to that question is no. Regulators do have discussions that may help advance medical device convergence within the International Medical Device Regulators Forum, or the IMDRF.
Thank you, Jill. We have another question for Dr. Lin Yao, and that question is, how long do you expect the E11A public comment period to last?
So we are hoping to have all comments back by November, December of this year, and then begin work on the comments.
Thank you. Let's see. Another question for you, Dr. Yao. What do you think are the most important new concepts presented in the E11A guide?
Yeah, thanks for that. I think that what we tried to do was, build on what was already available from FDA, EMA, and other areas. But I think the areas that are kind of new that I would really draw your attention to are the sections on extrapolation of safety, and then how to use modeling and simulation as well as specific study designs and statistical analysis methods as part of the pediatric extrapolation plan. I think those are really important additions, and additions that we heard stakeholders really wanted us to expand upon.
Thank you so much. We will move on to Dr. Alexey Khrenov. So we have a question for you, Dr. Khrenov. With the move from risk identification to hazard identification, are you not worried about drifting from ISO 31000? With the move for medical device quality management to be more aligned with the ISO 13485 approach, does this not set us up for difficulties?
Thank you. Very good question. We do not, as Jill already mentioned, medical devices, first of all, medical devices are not in the scope of ICH guidances. So they're going through different paths. Second, the framework as described in ISO 31000 is already slightly different, both in sort of general structure and terms used, for example, risk treatment, which is not used in the ICH-Q9 guidances. So the areas of applications are a little bit different because ICH-Q9 is focused on quality risk management for pharmaceuticals, obviously. So we already are not completely harmonized. And the third consideration here is that this revision was considered to be appropriate in terms of pure science, because what we identify first is the hazard, not the risk. And finally, it was done also to not contradict the existing text in Q9 guidance, because if you read the existing text of Q9 guidance, the risk assessment process mentioned in the text starts with the identification of the hazard. So all these factors basically made this revision necessary and appropriate for Q9, and other considerations like ISA differences, they're not that major, so they could not affect this revision. So we didn't consider them important enough to prevent this revision. Thank you.
Thank you. One more question for you. The FDA recently announced a new draft guidance for comments on benefit-risk considerations for product quality assessments. Is this draft guidance related to the revised ICH Q9 updates and therefore not expected to be finalized until after the ICH Q9 revisions are complete?
Not to my knowledge, while the guidance on risk-benefit is a CDER guidance, so it's limited to CDER. And of course, the ICH Q9 guidance is much more general applicability. They both touch on the risk. They talk about risk, however, on quite different topics and different angles. At least to my knowledge, there is no connection or harmony in finalizing the CDER guidance depending on ICH Q9. I'm not aware of that, so I don't think the revision of Q9 will impact this guidance, at least to my knowledge. Thank you.
Thank you. And I think we have time for one last question for you. When will the case studies become available for ICH Q9? This would better help understand the new revision.
Thank you. Actually, it's the same situation. The training materials will include case studies and example, and yes, I totally agree that this is one of the most important, the best tool to explain the revision topics for Q9, and we are working on the case studies. We are collecting case studies and working on them to package them in the training materials. And again, the answer is the same. They will be available on ICH websites as part of the training material package.
Thank you, Dr. Khrenov, and thank you to all of our speakers for your very informative and timely talks, and also for responding to the numerous questions that came in. We do have a few closing reminders.